Effect of nicotine on tetrahymena cells. Rationale. Our focus in this experiment was to determine the effect of various concentrations of nicotine on the cilia of tetrahymena cells. We were interested in this research due to its possible relations to the function of cilia in the human lung cells.
As we hop into our experimental design with the research question of how does the concentration of nicotine affect the number of food vacuoles formed in tetrahymena, we compared the independent variable of nicotine concentration at 0.0625%, 0.125%, 0.25%, and 0.375% against our dependent variable, the number of food vacuoles formed in tetrahymena. We knew that a concentration higher than 1% nicotine would immediately kill the cells, so we chose lower percentages of nicotine. We hypothesized that there would be fewer food vacuoles formed in the higher concentration of nicotine and more food vacuoles with the lower concentration of nicotine, since a higher concentration proved to kill the cells. We predicted a table similar to this would happen, where as the nicotine concentration increases, the food vacuole formed decreases.
Here's the data summary for our 0.0625% concentration of nicotine. Tetrahymena from a 72-hour culture and 2% proteose peptone were exposed to 0.0625% nicotine and 1% ink for 10 minutes, then fixed with glutaraldehyde and examined to count food vacuoles. The no-nicotine tetrahymena had an average of 4.37 vacuoles and a standard deviation of 2.15. The two datasets produced different outcomes, with one having an increase in average vacuole after the nicotine exposure, with 4.37 average vacuoles counted and a t-test p-value of 1.71 times 10 to the negative 6th, which is the figure 1. This p-value proves a significant difference between the nicotine-treated and untreated cells. The other dataset had no significant difference between the no-nicotine and nicotine cells, with an average vacuole count of 3.35 and a t-test p-value of 0.341, which is figure 2.
Using the same methods as previously, figure 3 shows that the number of food vacuoles formed by tetrahymena in a 0% nicotine solution and a 0.125% nicotine solution. A p-value of 0.228 was obtained, being greater than 0.05. It was concluded that there was no statistically significant difference between the tetrahymena that were not exposed to nicotine and the tetrahymena that were exposed to the 0.125% nicotine solution. This data supports to the null hypothesis of no significant change would be observed.
This is our data summary of 0.25% nicotine. Using the same methods as previous slides, figure 4 on the left shows the number of vacuoles created by tetrahymena with 0% nicotine and 0.25% nicotine. A p-value of 0.358 was obtained, indicating no statistical significant difference in tetrahymena behavior between 0% and 0.25% nicotine solutions. With a p-value of 0.358 being greater than 0.05, we determined that there was no statistical significant difference between the control 0 and the 0.25 nicotine solution when counting tetrahymena vacuoles. This meaning our data supports the null hypothesis that there would be no difference in the tetrahymena vacuoles creation in 0% and 0.25% nicotine solutions. This contradicts our alternative hypothesis that nicotine would cause difficulty in creating vacuoles in tetrahymena.
As we hop into the data summary of our 0.375% in concentration of nicotine, our figure 5 on the left, this displays the number of food vacuoles in tetrahymena formed in the control of the 2-peptone versus the number of food vacuoles formed in solution distilled in the 0.375 nicotine. The figure clearly shows that the nicotine did have a negative effect on the food vacuole formation, as we see more vacuoles formed in the control compared to the test substance, aka nicotine. We can also be sure that the results are not just random chances, which is gleaned from the p-value of 4.97023 times 10 to the negative 8th, which our original hypothesis stated that the higher concentration of nicotine, the fewer the food vacuoles will form. This data supports the hypothesis, and we can continue to theorize that the effect has to do with the destruction of cilia on the tetrahymena, but this theory would require further study.
Alright, so here we have all of the combined data from all of the previous experiments. Using the same methods as before, all of the t-tests were compared to all of the joint controls, giving a the 0.0625, p-value of 0.0143, a 0.125 p-value of 2.91 times 10 to the negative fourth, a 0.25 p-value of 0.49, and a 0.375% p-value, giving a 2.49 times 10 to the negative eighth. Using all of these values, 0.25 showed no statistical difference from the mean, with a p-value of greater than 0.5, supporting the null hypothesis. All other percentages having alternative hypothesis and show significant statistical evidence that the nicotine had an effect on the formation of vacuoles in the Tetrahymena.
Some of the limitations and errors that we incurred when we were conducting this experiment were as follows. The stock solution was discarded, and the 0.375% nicotine solution had only one set of data. Some of the cells were lysed. One of the controls had no tetrahymena cells several times. Tetrahymena vacuoles could be counted differently in each sample. When viewing cells, using different brightnesses can affect the viability of tetrahymena cells due to heat.
Some of the implications of this experiment. Our results show that there was a slight decrease in food vacuoles in the tetrahymena after they were exposed to the nicotine solution, meaning the tetrahymena could not digest the nicotine compared to a non-nicotine solution. Another result we found was that in all of the different concentrations of nicotine solution, many tetrahymena had lysed. This was not shown in the graph data because the lysed cells were not counted. The lysed cells show that with the nicotine solution, the tetrahymena was damaged beyond repair. This could be concluded because the cilia were damaged, thus showing that ciliated cells like lung cells are damaged when exposed to nicotine. The end. Thank you.